Hello everyone and welcome to the lecture 4 size exclusion chromatography in the online course Advances in Chromatography jointly organized by Sheth M and Science College Patan and DIPAM Foundation. Friends, when we talk about size exclusion chromatography, the name itself help us in understanding the theme behind it. It would be separating on the basis of size somewhere, or it would be related somewhere to biomolecules, biopolymers, polymers, macromolecules, etc. So let us take a deep insight into it. Whenever we say size exclusion chromatography, it is based on separation of molecules on the basis of size. It means that separation of biomolecules in aqueous system is equivalent to gel filtration chromatography. Similarly, separation of organic polymers in non-aqueous system is equal to gel permeation chromatography. Remember very well over here, if we compare it with few of the other chromatography techniques, over there it was a binding between the stationary phase and the analyte, where we had bonded phase chromatography, where we had HPLC, where we had GC, MPLC, LPLC, UPLC, etc. But over here in size exclusion chromatography, you do not have binding. It is just an attraction. It is not at all an binding. And even attraction, not in the terms of force, but on the basis of size. And henceforth, it is very written in this slide that binding with the stationary phase does not occur. Molecules to be separated percolate as per their size. So it is just percolation, passing on of different sized molecules into a column. Size exclusion chromatography, if we talk about its working principle, separation of component based on molecular size, differential exclusion on passing through stationary phase at different pore size. There are various columns available in market where in a single column the stationary phase of different size pores is filled in a chronological order, and differently exclusion of the differently sized molecules take place. Over here, what do I mean by exclusion will be coming up in the next slide. High retention of biomolecular activity, which is equivalent to the moderate interaction with the components. Now here is the question. Why do we say it to be a moderate interaction with the components? Because over here it is mentioned binding with the stationary phase does not occur. So if the binding with the stationary phase does not occur, there is a very little interaction between the stationary phase and your sample. And as the interaction is very less, it will not be much retended into the column. So it is considered over here as a moderate interaction with the components. Working principle can be explained in a very different way. In size exclusion chromatography, the column filled with gel particles or porous stationary phase is equilibrated with mobile phase. It means, first of all, when the mobile phase is passed through the stationary phase column, the gel particles get equilibrated, get saturated with the mobile phase. The introduction of sample percolates the components with the mobile phase. So with the flow of the mobile phase, the sample keeps on coming down and down and down. Now comes the science. The large molecule do not enter into the pore and are excluded through the vacant space not occupied by particles. So when from the top of the column the mixture is passed along with the mobile phase, the particle or the component in the mixture with the larger size do just pass through the pores, do not enter into the pores, whereas the small size particles do enter into the pores. So the larger size particles which are excluded will just go away and not at all enter into this tiny pored stationary phase. So it can be written as the large molecules do not enter into the pores and are excluded through the vacant space not occupied by the particles, whereas the smaller molecules are distributed in the mobile phase and molecular sieves, means they are trapped into the stationary phase. This results in the fast movement of the larger particles as compared to slow. Now this is important. Why we say that the particles with the larger size move fast? Because they are not trapped in the stationary phase. If you have a column, the larger particles do not fit into the column, they will run down. But the smaller particles will fit inside and will be retarded more. So in size exclusion chromatography, you can say that the larger particles are moving down, whereas the smaller particles are remaining inside the column and trapped. So the larger particles are excluded out quickly. This is the base when we try to explain size exclusion chromatography. What is very important to understand over here? Two words: exclusion and inclusion. The exclusion and inclusion of particles depends on the size range chosen. As per the parameters, all the components having size equal to or larger than the critical mass behave identically and are excluded from the column. I mean to say, if the pore size of my stationary phase filled in the column is 10 micrometer, 20 micrometer, or let us assume 100 micrometer, or even let us make it easy 100x, then all the particles having size larger than 100x or 100x will be eliminated, will not pass, but will not get trapped. But the component, the particle having size less than that will get trapped inside. It means the larger particles or equal sized particles equal to the stationary phase pores will not get trapped, will be excluded and thrown away with the mobile phase, whereas the particles having lesser size will get trapped. These components are eluted as excluded of the column. The particles having size lower than the critical mass are captured by pores and are eluted as included volume. Friends, over here, 5 environment science analytical chemistry, molecular exclusion chromatography. Please eliminate this section. It is not included. It is just cut paste error that has been done over here when I copied it from the material to the PPT. For us, it is these components are eluted as excluded volume. These components are eluted as excluded volume. And the one that gets trapped is called included volume. So these two terms are very important. Those components from the mixture which do not get entrapped into the pores and are thrown away with the mobile phase are called excluded volume, and the components that get trapped are called included volume. Size exclusion chromatography has a very important aspect of separation mechanism. There are two methods. Method 1, distribution between two liquid phases, and method 2, competing for vacant site. Let us start with the method 1, distribution between two liquid phases. In size exclusion chromatography, the components are distributed between two liquid. One, liquid present inside the pore or gel, and the liquid present outside. Now, liquid present inside generally is considered as a stationary phase. Liquid present outside is generally considered as a mobile phase. So, it's some liquid-liquid interaction type of partition. The distribution follows steric exclusion mechanism. What does it mean? It means small components can enter into small and large pore both. However, large component can enter into only large pore. Therefore, different pore fractions are available to the molecules of different size. This means the distribution coefficient of component with different size differ in two liquid phases. Means the distribution of differently sized particles is distributed between stationary phase liquid and mobile phase liquid. And mind well, stationary phase liquid fundamentally is a gel. If you go back to my old lectures, I have very clearly mentioned solid coated, liquid coated on a solid or a gel form will be considered as a liquid stationary phase. It exists and it is possible. Now, this distribution between two liquid phases can be explained. See how V total is the total volume, where Vg is the volume occupied by the solid matrices of the gel, Vi is the volume of the solvent held in the pores, V0 is the free volume inside the gel particles. So this is just the equation and the terms related to it. The diffusion equilibrium and the retention time of the component is given by Vr is equal to V initial plus KdV initial, where Kd is equal to V initial divided by V total, where V initial is the accessible pore volume which can be assessed, and V total is the total pore volume. This is just a mathematical explanation, but our aim should be a thorough understanding of this mechanism. Mechanism with a different view, competing for vacant sites. In secondary exclusion mechanism, the small molecules diffuse rapidly into the pores of the gel, while the larger molecules compete for vacant sites. Small molecules diffuse rapidly into the pores, they go into the pores, while the large molecules compete for the vacant site, they go towards the bottom of the column. The larger component move down and find relatively more occupied site downwards. This results in the separation of smaller components collected at the upper end and large component collected at the larger end, lower end. Friends, be very clear. At the upper end, smaller components, at the lower end, larger components. This is the main idea for size exclusion chromatography. You can see in this picture. Over here, it's a mixture of two different components, the red and the green. These are the pores. You have a red color, bigger size molecule, and green color, smaller size molecule. With the passage of mobile phase, the green color molecules are trapped inside, but the red color molecules come out first. It means the larger size particles are excluded first. Sometimes they come out with a multiple choice question that in size exclusion chromatography, if the size of A component is larger than B component, which will be excluded first? A will be excluded first. So this particular picture taken from Google helps us in determining the exact separation mechanism when we are competing for the vacant sites. So be very clear when we are just discussing this particular slide. Moving ahead, instrumentation. Size exclusion chromatography has a similar kind of instrumentation with the only change in the column material, stationary phase, that is the packing material. It has a solvent supply, that's the solvent reservoir, solvent delivery system which includes high pressure pumps. You have an injector, you have a sample, you have a column, you have a detector, followed by a data system. So the same simple instrumentation with no major change. But the stationary phase that is filled inside this stationary phase, filled inside this column is very important. The stationary phase used in size exclusion chromatography is semi-permeable, means it will allow molecules to pass through. You must know this definition. Permeable, non-permeable and semi-permeable. Non-permeable, not allowed. Permeable, totally allowed. And semi-permeable, partially allowed. If you go back and think of osmosis, you have semi-permeable over there. And it is made up of cross-linked polymer with porous nature. This porous nature makes the particle to pass through it. It has a well-defined pores and the size of the pores is controlled during cross-linking. Remember very well, higher the cross-linking, smaller the pores, lesser the cross-linking, larger the pores. So these small pores are used for purification of protein, desalting of protein. Medium pores are used for separation of small size protein and large pores are used for biological samples. So these are the three important categories: small pores, medium pores, large pores. If you have stationary phase made up of small pores, desalting of protein is possible. But if you are interested in separating small size protein, you must have medium pores. And if you want to separate biological samples, you must have large pores. Let us go into detail of it. Dextran polyacrylamide is one of the very popular commercial stationary phase used in size exclusion chromatography. It is uniform size and shape, available in various different sizes and shapes of the pores. It is non-reactive, propertized, so it does not react with the sample or even with the mobile phase, inert, and is mechanically stable. Because in this HPLC type of size exclusion chromatography, you have high pressure coming up from the pumps and it should not get mechanically disturbed. Due to pressure, the size of the balls, small, should not change because these gels are small circular balls. Apart from this, dextran polyacrylamide, you have three different commercially available stationary phase: dextran alone, polyacrylamide alone and agarose. Number 1, dextran. This gel is homopolysaccharide of glucose and can be prepared by varying the cross-linking to control the pore size. It is available in dry beads, circular beads, under the trade name Cephadax, which swell in presence of water. So remember very well, Cephadax, fundamentally dextran, a homopolysaccharide of glucose, swells in presence of water, so it is hydrophilic. Mainly it is used for the separation of small peptides and globular proteins with small to average molecular mass. So remember this very well. Dextran, made up of homopolysaccharide of glucose, is used to separate globular proteins. When we talk of polyacrylamide, it is also a gel and can be prepared by cross-linking N-methyl, N-methylene by or bisacrylamide. So you should remember this. When we write bisacrylamide, fundamentally it's organic. Its separation tendency is same as dextran and is available in various sizes. It is available as biogel P. This biogel P is a trade name. Agarose, it is a gel compound of linear polymer of D-galactose and 3,6 anhydro-1-galactose. It shows dual nature. What it means? It is dissolved in boiling water and is converted to gel in cold water. So when you have boiling water, it becomes a solution. But when you have a cold water, it becomes a gel. Its pore size is larger as compared to the gel discussed before. So the pore size are larger compared to dextran and polyacrylamide. It is used for the separation of large molecules of DNA and sometimes for globular proteins of larger size. So friends, this is the role of stationary phase, which according to me is the key factor when it comes to size exclusion chromatography. Mobile phase is normal, what we have seen earlier. The mobile phase is selected on the basis of the nature of components to be separated. Most commonly THF, that is tetrahydrofuran, chloroform, toluene, dimethyl formamide and mixed solvents are used to separate various components. In case of proteins and polysaccharides, aqueous buffer are used as mobile phase. Aqueous buffers. Phosphate buffer is an aqueous buffer. The solvent should be able to dissolve the components of a mixture and must be inert towards various components. Keep very well note that your mobile phase should not react with your sample. The detectable properties of the solvent must be different from the sample component. It should not be corrosive. Otherwise, it will corrode the column, which is made up of metal. Generally, stainless steel is used as an anti-corrode material. Before using a mobile phase for analysis, high purity solvent is passed through a filter paper of 5 microns, zero point 0.5 microns to remove the dust and insoluble salts, which helps in getting relieved from the choking up of the columns. The columns of various length and diameter are available commercially. However, small columns are recommended to save the time and solvent. Why? Because the smaller the column, the smaller the radii. The smaller the radii, you will have the lesser the volume. The lesser the volume, the wastage of the mobile phase or the use of mobile phase will be less. The particle size selected depending upon the analyte. If you are interested in separating two components of a very similar properties, particle size has to be smaller, which will increase the number of theoretical plates. But the two components to be separated are fair different in their properties. You need to take a little larger particle size, resulting in the decrement of the number of theoretical plates. Generally, particles with 5 mm size are selected, but small size particles are more sensitive towards contamination because the molecules get trapped inside and at a larger time span spent inside results in a reaction. For high molecular weight, large particles is recommended because small size of particle may damage the large size analytes. The porosity of the column is also responsible for the separation of the components. Before operation, a column is saturated with the mobile phase and handled with care. Friends, somewhere while typing or cut pasting it from the material, the PDF, the title is getting repeated, that is environment science analytical chemistry, what I teach at my university, having the topic ion exclusion chromatography or molecular exclusion chromatography. also known as size exclusion chromatography. So avoid that line wherever you find it. We'll try to remove it from the PDF that we regularly share with you. Size exclusion chromatography utilizing high pressure pumps and detectors. The pumps that are used to maintain the constant flow rate during the analysis. A change in the flow rate may cause error in the result. The inline filters prevent the entry of particles and save the valves and pump from damage. These are the rational things that we have earlier done, so I'm not taking much time in that. But the main theme was size exclusion. Detectors used in SEC are either concentration sensitive detectors, RI detectors, UV detectors, or evaporative light scattering detectors. Also remember that viscosity detectors are very much useful in size exclusion chromatography. Its applications involve purification of proteins and biomolecules, separation of sugar, peptide, rubber, and others, protein ligand binding studies, protein folding studies, copolymerization study, and relative molecular mass determination. So, friends, a very short topic, size exclusion chromatography, comes to an end over here. Thank you. Along with this video, in the description, I am keeping 3 more links. You are requested to go through all these 4 techniques, which somehow we would not include in the basic of chromatography online series, which have been included over here. And then after, we will continue with some other important aspects of chromatography. Thank you once again.